Hello, students and participants for MOOC course, Introduction to Proteogenomics. Welcome to week 8. I hope you're able to catch up on the previous week's lectures and assignment. And now you're ready to take some new lectures and new learning from this week. As every week, we are going to give you some more reference materials in the video description. Also, please utilize the Skype meeting, which we conduct every Saturday, so that you can clarify your doubts. We have also made a Telegram group. You can utilize more interaction and more discussion in that Telegram group. So please do join that group as well. Coming to this week, the first lecture will be about Human Protein Atlas project. This is a big initiative from the Uppsala University and Swedish groups, which is the large program to look at more antibody-based characterization of the proteins and creating the atlas, looking at the entire proteome maps, as well as looking at now the cellular and the subcellular context. So Dr. Ponting, he's going to give you at least some overview of the Human Protein Atlas project, and also give you idea that how different type of programs are being coordinated in HPA, both antibody-based and some of the mass spec-based work. In the next lecture, he's going to give you some more detail about the Cell Atlas project, Human Protein Atlas Brain project, and you will actually realize that how crucial it is to link these data sets with the pathology atlas, where you're going to look at the mRNA and the protein expression data set of different cancer. So some of these initiatives are really important because they provide you a resource and the availability of so much information, which you can use to know more about your proteins of interest. You can go to the Human Protein Atlas website and look at the different proteins of your interest, where they are localized, what is known for them in different disease contexts, what is their subcellular localization. All of these information could be understood from this particular portal. Temporarily, this portal is, I think, down right now because of they're doing some maintenance. But please keep track, keep an eye on this website, and whenever it is up and running again, you will be able to get a lot more wealth of resource and information from this particular Human Protein Atlas website and this initiative. In the next lecture from the same HPA team, Dr. Joachim, he's going to talk to you about Human Plasma Proteome projects. Again, the Human Protein Atlas is a very large initiative. Different teams are working on different aspects of the different tissue and blood project. And this initiative with Dr. Jochen and his team from KTH Royal Institute of Technology is going to give you an idea that what is initiatives happening with the plasma proteomics. Plasma proteomics, as you can understand, is very challenging given the abundance of different proteins in the human plasma. How to remove the abundant proteins and still identify the low abundant biomarkers, low abundant proteins, and look at the different methodology for the discovery work, validation work, integration with other type of clinical usage. So Dr. Joachim is going to talk to you about all of them, and also giving you idea for the Atlas Antibody project as well as the biobanks initiatives. All of these are really going to make you updated about the best initiatives, the most recent initiatives happening in the field of proteogenomics and proteomics research. From the previous weeks, I hope you are able to catch up to the large initiative of CPTAC and the NCI programs. And now we are introducing you to new programs of Human Protein Atlas-based projects. But finally, in this week, you will be intrigued and stimulated with two interesting lectures by Dr. Sachin Jadhav. He is one of the clinicians who has given you perspective about looking at the clinical problems, and as a clinician, what kind of limitations he sees for the research, what kind of gaps he sees. And then in the following lecture, he's going to give you some very interesting examples of looking at different type of mutation profiling and how best they have gone about investigating these clinical problems. So on one hand, you are going to get the technology aspect of the of the proteomics and proteogenomics, but also let's be realistic that how some of these technologies which we are talking and working on how they're going to be impactful for the clinicians and people who are going to make use of them for the patient outcomes. So what is their viewpoints, what are the challenges which they face, and how they're still trying to overcome some of these problems and making the progress further. So these lectures are going to be very stimulating for you, and I hope this week is going to keep you highly motivated as well as less busy as compared to previous week when you had too much technical software to go through. Here you have more good information to absorb. All right, that's all for this week. I hope you'll be able to finish your assignments well, and please do ask more questions and interact more actively with us. Thank you.